This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 83 for May 21st, 2010. Hey everybody, I'm Vincent Racaniello, and this is TWiV, your weekly dose of virology. And today we have a great crew, starting with me here on the East Coast and moving to Western Massachusetts, where Alan Dove is back.
Yes, after a 2-week hiatus, I am back on TWiV.
Good to have you back. Seems like forever.
Yeah, it seemed like ages.
Yeah, well, that's good. You're here, and Dick Despommier is gone again. Oh. Someday we'll have the original crew back together. So moving down the eastern coast of the U.S. to north central Florida is Rich Condit.
Hello, everybody.
How you
Great
doing?
to be here.
I know, it's 90 degrees there, right?
88. Close enough.
Great.
And humid. It was it's humid. It's getting there. We're getting into summer here.
All right. And I'm really happy to have joining us today across the country from San Francisco area, from This Week in Science and Dr. Kiki's Science Hour, Dr. Kirsten Sanford. Welcome.
Thank you. It's great to be here.
Yeah, good to have you. We thought it would be great to have you join us, talk about your career, post-PhD career, and then join the general chat on virology. Thought you might like to do that. So thanks for coming on.
Yeah, I'm going to enjoy it a lot. This will be fun.
So I thought we'd start by talking about you, which is probably something that you're not used to, but we have a lot of early career scientists listening to TWiV, and we always like to tell them what different kinds of careers they could have, and you've certainly had an unusual one. And so I thought we could start out by just talking about your early days in science. You have a PhD. Not an old one either, 2006, right?
Yeah, end of 2006.
You're newly minted, really.
I am. I'm a little baby PhD.
So at some point in those years, you decided you didn't want to do a traditional research career?
Yeah, so what happened in my grad school career is before I started grad school, actually, or right around the same time as I was starting my PhD studies, I started doing This Week in Science, just as a radio show out of UC Davis. And I knew I had done radio earlier, years before, and approached the public affairs director at the radio station and said, hey, you don't have a science show. Maybe you should, you could have one. I've got this idea. And they said, okay, let's give it a try. And so I started doing This Week in Science, and then started my grad studies, and everything worked in parallel. My working in media on a small scale, and also moving forward in science. And about halfway through grad school, I had finished my orals, and so my coursework was over, and I was getting ready to dig into just the academic studies and the experimental studies. And my advisor decided, she had announced to the lab that she was moving back to England, where she had originally come from, and she wasn't taking anybody with her. And so there I was. And I went, what do I do? I don't have a lab. I'm not going to have a place to work. Where am I going to go? Nobody's doing work like this at UC Davis. So what am I going to do? And so, I actually took a year off, a leave of absence from school for a year to kind of put my thoughts together. And I, in doing so, left the radio show. I stopped doing twists, and I left it to a couple of guys to keep carrying the torch while I was gone. And I realized that the thing I missed the most was the radio show. I missed talking about the science. and I thought about it. I was like, wow, the stuff that I really, really love about science is the communication and the ideas, and the being able to spread ideas is, to me, just one of the best things in the world. And so I finished my leave of absence. I found another lab to work in at UC Davis. And so I went back to finish my grad school career with the intention of moving into the communications field.
So you were really into communicating before you went to grad school then?
I was, and it's really funny that I never really put it all together until, actually just very recently, a couple of years ago, that it was ever since I was little, I've been doing media type stuff, communications type things, but I had it in my head that I need to do science. Because I've always loved science. I've always, ever since I was little, I was doing science fairs too. Always asking questions and always doing the scientific thing. But and so I just thought that was what I was supposed to do, go into academics, go, the professor path, whatever it happened to be. And it was all of a sudden this moment in time that kind of forced me into realizing, wow, the communications and science don't have to be these two separate things.
Well, traditionally they are, right? Because scientists do their thing and they don't talk very much about it outside of their colleagues.
Right.
So I'm interested that having made this discovery in your leave of absence, you actually went back and completed your PhD, because that's not light duty. Where'd you find the motivation to do that? How did that work?
I asked a lot of people what I should do. I contacted science journalists like Joe Palca at Science Friday and NPR, and a number of different people who were working in the field to see what they thought, what their advice might be. And a number of them said, don't worry about it. You don't need a degree to be a science writer. And a number of them said, maybe get a master's degree, and, then maybe go into a science journalism program. And then a couple of other people said, yeah, go ahead, do the grad school, do whatever you want to do. So basically what the advice came down to is, if you have a background in science and an interest in science and an ability to write, you don't need to go through the grad school experience. But I kept thinking to myself, I really like science, and I really wanted the experience of fully going through the dissertation challenge. I wanted to know what it was like to publish a paper. I wanted to know what it was like to, try studies and have them fail. I wanted to continue along the scientific path up to a point where I could actually have something to show for it in terms of credibility. And basically, yeah.
Do you think that's helped?
So how would you advise people, having had this experience, what would be your advice to somebody in the same situation? I think, any grad masters or PhD, PhD is a long haul. In all, it ended up taking me 6 years to finish, with the leave of absence. But it's not a short thing to do. And so if you're going to do it, you should really know why you're doing it. And have a goal in mind at the end of the road. Because if you don't have a goal in mind, no matter what, it's going to be a struggle. And my goal of knowing that I wanted to use what I learned in grad school, to be able to help me in my science communications career, and move me forward in that, knowing that there was something at the end of the road for me, it kept me going. And I was able to, through the hard times and the times when I wanted to quit everything, I was like, I can do this. I have a goal in mind. And so that's really the big thing. But also you need to make sure you have an advisor who accepts the fact that you're not going to be focusing on an academic career and a professorial career, because there are still many people within academia who will not really assist a grad student who isn't going to be focused on continuing to publish papers and make them look good.
Yeah, no. I know that situation. In fact, Alan Dove came to me shortly before he was going to get his PhD. So Alan, who's with us, did his PhD in my lab. And he
said, I can't empathize with any of this.
He said, I don't want to do.
Actually, Vince, you forgot the setup to this story.
Okay, tell me.
Michael Bouchard, who was in the lab at the same time, sat right across from me, and he was finishing up his PhD and was kind of about a year ahead of me, and he was thinking about switching into something like medical school, and he went to Vince's office and told Vince this, and he came back to the lab and he said, wow, that didn't go well. Vince heard me out, and then he looked me in the eye and he said, is that what I trained you for?
Oh dear.
And so I knew I was going to have a similar conversation with him, and I really didn't look forward to it, because by that point I'd made up my mind I was going into science journalism. And so a couple of weeks later, I got up my nerve and I went into Vince's office and I said, look, I'm obviously going to reach a point in a couple of years. Hopefully I'm going to be finishing this up, maybe a year. And I am not interested in doing a postdoc. I'm planning to go into science journalism. And Vince looked at me and he nodded and said, yeah, that's probably a good idea.
Oh dear. All this coming back. Yes.
That's really funny.
Well, since you guys, there have been many others who've gone on to all kinds of different careers. So I thoroughly have now understood, or a long time ago understood, that that's fine. And
your advice was absolutely correct. Michael, as we just heard a few weeks ago, has gone on to do great things as a basic researcher, and I, of course, have been happy as a science journalist.
Yeah, you've done very well yourself too. Absolutely. No,
I've adopted the attitude that, your role as a mentor is not to tell somebody what to do, but to help them find their way, whatever that might be.
I think
there's lots of different ways.
I think that's very appropriate. So, I mean, in addition to advising the grad students who might be, or postdocs even, who might be listening to this, we're also advising the advisors.
Yeah.
Absolutely. No, you have to be. You're not
going to get 100% buy-in to this from the advisors.
You're absolutely right. No. I think that's a great one, Rich. I like that. So you got your PhD and you were doing TWiV.
I was, yes.
Here you are out the door. What did you do?
Out the door, I made sure that I had a couple of papers in the pipeline, and everything was nice, so I could close that chapter of my life. And I started making contacts in the new media area. So I ended up meeting Alex Lindsay from Pixel Core, and he's involved with MacBreak Weekly, I think. Mm-hmm. MacBreak Weekly, and Leo Laporte and a number of other individuals in the new media sphere. And we met at a podcasting conference. So I started going to conferences to try and meet people. And we met at this conference, and he said, "Oh, you do This Week in Science. You do science. I like that. I have an idea for a science show." And so we ended up collaborating on a video program called Food Science that it was pretty popular when it came out, and it was a short run because we didn't have a lot of funding to keep it going, unfortunately. But we had a lot of fun demonstrating kitchen science and a whole bunch of things that happen dealing with food and with cleaning solvents and all sorts of stuff in the kitchen. And Pixel Core is known for their motion graphics and their illustrative capabilities. And so the graphics that we had were all nice and sparkly and awesome. So that was really fun. And from that, I was able to, doing that kind of opened a door for me to get into another program on a network called Revision 3, this show called Pop Siren, in which I did a segment called Rad Science. And again, it was kind of a demonstrative segment talking about different aspects of science and DIY projects that you could do at home. And so it's just been kind of step by step, meeting more people within the field and networking and getting to know people. And through the stuff I've done with these groups, I eventually was able to make contacts with the Science Channel and have made other contacts within the Discovery Network and National Geographic. And so people outside of just the internet sphere, I'm now moving into the larger media field, and I've done some work on the Science Channel as well.
So I guess the an important question is, can you make a living doing this?
You took the words right out of my mouth.
You can make a living doing this. It's very difficult. I mean, it's definitely been a lot of bootstrapping, and getting one project is not the kind of thing that's going to keep you in house and home. It's getting a number of projects and making sure that you have a lot of work coming in. So it's a lot of work to make sure that you have the contacts, to have the projects, to be able to. I do consulting and contracting on the side, so there are always a lot of I guess balls in the air for the stuff that I'm doing. It's I've also had there are always the possibilities of developing new projects, and if you work with a nonprofit organization or even through a university or other kind of institution, there's grant funding. There are all sorts of ways to make the money happen. It's just you have to put the time and the work into actually making that happen.
So how do you do a very broad cut of science?
Yeah.
You cover everything. How do you do that?
I like everything, so that makes it easy and fun. I have to read a lot. I have to keep my eyes on all sorts of journals and media outlets and find out what's going on. And I've been doing it now. I started This Week in Science 10 years ago. So, even though I was focusing very, very directly during grad school, I was also paying attention to a lot of different fields, and I love interviewing different people. And so even though I don't know the answers to things, I can ask questions. And so I try and provide a doorway or a path for other scientists to be able to talk about what they do and explain it to people. So I think it's just having that curiosity of wanting to know how everything works. And I think that was what grad school never quite gave me, because I had to focus too much on one little tiny thing. And I was like, but there's all this other stuff out there.
Yeah. Well, I think that people have to realize that you work at it, that you just don't sit down and talk about all kinds of science.
Oh, no. Yeah. There is work. There's work involved in all of it. And, it's fun. I enjoy it, but I have to do background research if I'm going to talk about something. If I'm talking about dark matter, I have to make sure I have a little bit of an idea about what dark matter is. Sure. I don't have to be an expert in it, but I have to know enough about it to be able to figure out, what questions I should be asking.
So do you think
Go ahead.
Sorry.
I would think it would get easier as you went along, because you pick up more stuff and stuff comes around again. So your background increases as time goes on.
Right. And there are certain stories that seem to pop up over and over again. Like, I guess last week it was the Neanderthals and humans share DNA. Woo! Like, exciting. But that's been something that we've been following on This Week in Science for years and years and years. And so, that story comes up, and we don't have to do a lot of background research because we've been talking about it. Other things having to do with Saturn and the Cassini mission, talking about Saturn's rings and other things. Again, we've been talking about it for years. So there are certain topics that just are a constant, and so you're constantly dipping in. And the neat thing is, though, because science is always moving forward, you have this growing base of information that's always changing in a slight way.
The way I look at it is, the more you learn, the more you realize what you don't know.
Yes.
And so that becomes a problem.
Yeah.
So recently you started a QA, a live question and answer. Is that right?
I did, and it's been so much fun. It's just my science chat, and I'm doing it on Justin TV on Fridays at noon Pacific time. So right after we're done recording this, I'm going to hop over to Justin TV and do that. But it's been really fun. it's a different audience because a lot of the outlets that I've had so far, I mean, especially with podcasting as well, it's very niche, and the people who are very interested in what you're talking about, they're going to find you and search you out. And so I'm interested in trying to find ways to reach people who don't necessarily know they're interested in science. And so how do I get out of the very, just having the science bubble? How do I get out of this science audience bubble? I love all the science audience people. They're fantastic, but how do I get new people to be part of that audience? And Justin TV is I think a much broader cross section of people. And I started doing this chat, and it's just this little chat room. I have a webcam and my microphone, and so I talk, and there's just this little chat room. I read questions from the chat room and then answer them. And it's amazing. I have like over 1,000 people in the chat room, and these questions coming at me like a mile a minute. And
can you answer them all, or do you have some you can't answer?
Oh, there are some I cannot answer, and I say I can't answer them. And I try and tell people to email me so that I can remember to look up answers and try and come to the show with an answer the next week. I've only done it for 3 weeks now, so this is my third or fourth week, and so I'm still getting into it and figuring out the format. But the thing that really struck me was just the response and how many questions people have. I don't know how many opportunities people have to just ask questions and have them answered in an honest way.
Well, certainly not.
We need to put a link to this in the show notes, because I'm gonna ask you a bunch of questions.
Yeah, you should do that. It's a good idea. I think that people don't always ask the right people their questions, right?
Right.
They don't ask someone who understands science, in which you do in your case, but they may ask their neighbor or someone they find online who doesn't know the answer, and that's how all misconceptions get propagated. So this is good. And in fact, we've been toying with the idea of doing it here while we do TWiV, is have people send in questions. And so at some point we might do that. But I think just from our response, having the email, we do a lot of email questions on TWiV. It's fabulous. Yeah. And I think for me, it's the most satisfying part, to have people send in questions and then we answer them. I just love that part of this whole thing.
Well, there's so much of the interactivity, and so much of the reason why I do what I do, and I'm sure it's why you do what you do as well, is you want communication. You want to foster people asking questions, because that fosters education and the growth of knowledge and understanding. So, I mean, in all, it's the interactivity that really makes a difference. If you're just pushing information out at people, I mean, what are you doing?
Sure. Yeah, no, I think we will want to teach people, and we have a certain expertise that is rather rare, so we figured let's tell people about it, because people don't usually have the opportunity to ask virologists about what's going on. So
absolutely,
it hits home. And I think for any branch of science, you could argue the same thing. And you would always find people who would have questions for you. So I think that's a really good way to do it. I like the interactivity part. It's not something you don't do on Dr. Kiki's Science Hour, for example, right?
Not so much. There is a chat room that I keep an eye on, and occasionally, if there's a really good question from the chat room, I'll incorporate it into the show. But it's not something that I've really done very well yet. It's something that we tried early on, but then it just didn't quite work. Like, I don't know.
Having been on a few TWiT podcasts, I find the chat room very difficult to parse, because here you're having a conversation and then hundreds and hundreds of questions are spinning by, and it's very difficult. Whereas maybe if you had a part of the show dedicated to just answering questions,
right,
not try to have a conversation with someone else at the same time, it might be easier. So not too long ago, you started Dr. Kiki's Science Hour on the TWiT network, and I suppose that furthered the reach of your work, right? Yeah.
So the Science Hour has, I think there's been a little bit of overlap between the This Week in Science audience and the This Week in Tech audience, but in all, I've gotten a bunch of new people who have started emailing me and saying, "Oh, I'd love it if you do a show about this. Can you interview somebody about oil? There's this huge thing going on in the Gulf. Can we talk about this natural resource? Or can you get somebody to talk about quantum dynamics? Or all sorts of interesting questions."
Now people are full of them. It's great. They could certainly make shows for you forever.
I know. Right. I just have to. The challenge though is making sure that that the audience grows and that the audience is happy and that you're continuing to create a show that is doing a service for those people.
So what's in the future for you in science education?
In the future, I have a couple of show ideas I'm toying with right now. I have a little idea in the back of my head that I don't know. I'm playing with an idea. I'm going to keep it quiet for right now, but just a little tiny tease. Tease. I'm teasing. Yes. I'm still talking with the Science Channel. We'll see if there's more work there in the future. And there are ideas floating around This Week in Tech and that network. We'll see where it's going. A lot of what I'm looking at right now, I'm going to see where the Justin TV stuff is going, because I really enjoy what I'm doing there on Fridays, and if that could turn into something that I could do more often.
Mm-hmm.
Yeah.
How about? Are you interested in eventually going on the national television network scene and reaching a lot of people that way?
I would love to. I would absolutely love to. Yeah, if I could, if I could make the right contacts and get the right spot. I mean, it might be doing something similar to what Michio Kaku or Neil deGrasse Tyson does, what they do now in, talking about various science topics on Fox News or CNN or whenever they're invited in to answer questions about the new big science issue that the media is reporting on. That could work. I would also really love to have some kind of science program of my own. It would be a dream.
Keep working on it. You could do it. Just like you went back and got your PhD. You can do it. I must tell you, I first found you on Mac Break or Mac Break Weekly, I think.
Okay.
You were doing some interviews at a trade show.
Yeah, I've been, for the last few years during Macworld, for Mac Break, I've done some interviews. I run around and find sciencey things at the trade show to be able to report on.
So then I found your science work and found TWiS and eventually the Science Hour. And at the very beginning of TWiV, I was talking about science podcasts, and I think I had mentioned Futures in Biotech, one of the first science podcasts, and immediately some listener wrote, "That's not true. TWiS has been around since the Precambrian era."
That's true. Yeah.
So you have defenders out there everywhere. Anyway,
that's good.
Anyway, it's great. I really enjoy what you do, and I hope
Thank you.
I hope the listeners enjoy it. If you have questions, we'll give you some links later on where to reach you.
Absolutely.
That's terrific. You've really accomplished a great deal, and it's wonderful, and good luck.
Thank you so much. Thank you. I appreciate it.
And for those PhD students who want to do something different, I think you're a great role model, and they should pursue their dreams. It can be done.
It can be done. It's just, you put your mind to it, you find whatever it is that is your dream, that's your passion, and you go after it, and you can do it. And there are things outside of academia. It's true. And you can use your degree for so much.
Absolutely. No question about it. Look at Alan Dove.
And
yes.
Good job. Well, and good job, carving out a niche in new media as it was developing. I mean, I started with old media and have gradually morphed into the newer stuff. But you jumped on it early, and that's great.
Well, when you first went out, there wasn't new media, Alan. That was the reason. Maybe you would have jumped on it too.
Exactly. To me, new media was a dial-up modem.
That's funny. Yeah, I think there is a certain amount of what I've done in terms of being being on the bleeding edge and being, in the right place at the right time, as we were just keeping up with the changes in the technology. So as websites came through and MP3s and then podcasting, like we just TWiS just kind of kept doing the newest thing and trying to do something different. And it really, I think paying attention to the technology and really trying to grow with it has made a difference.
I try and use this in my classes. I try and get them to use the podcasts, and there's so much potential for teaching in this new media. It hasn't even been tapped yet.
Oh,
and that's one of the things I'm very interested in doing. And the TWiT network with this incredible 4-way Skype video and audio, I think this is perfect for teaching a class. And someday I want to just figure out how to do that.
Yeah. I think that's a great thing to try and figure out. And there are lots of people who are, working on those, those broadcasting techniques and technologies. So there are lots of people to talk to.
Right. Well, I'd like you to stick around and talk about some viruses with us.
Cool.
We have a couple of interesting stories today. But before we do that, a word from the sponsors of this episode of TWiV, Data Robotics. And by the way, Kirsten, Mark Fuccio said to say hello to you.
He's wonderful. I am a Drobo user.
Yes.
And a fan of Data Robotics. Yeah, a very big fan.
He said, have a conversation with Kirsten instead of just talking in a monotone about the product. Okay, so we will. Drobo, the makers of this wonderful storage solution for your computer. And these are wonderful black boxes that you slide drives in and have totally protected and huge amounts of storage. And I've got 3 of them. I started out with the original Drobo 4-bay model. Now I have 2 other 5-bay models. Here at the lab, I put all our data on it. I put all my podcasts, all my lectures, all the videos of my lectures, and I feel that it's all protected, and it's great. What do you use a Drobo for, Kirsten?
I use my Drobo for storage of my media. I back up all of I probably have almost all, not quite all 10 years, but almost 10 years of This Week in Science in my Drobo. And I've got all the video that I produce that I need to store. And when I'm working on editing or any of that sort of thing, my Drobo is right there. I use it for not just storage, but also accessing as a second drive for my computer for video and audio editing. It's pretty great.
Which model do you have?
The little one.
The four drive one, yeah. The little one. Hey, you got that, Mark? The little one. That's good.
This is my remembering the model numbers and whatever.
It's a lot.
It's the cute one. It fits on my desk, and I like that.
It's pretty. It's black. It's got green and red and orange lights. It's really very nice if you like that sort of thing. But
you can enter the contest and maybe get an upgrade.
Yeah, that's right. We have a contest ongoing to win a Drobo. Actually, they gave us 3 to give away, and we're on the 3rd one. The first one was just to actually subscribe to TWiV and then tweet it, and then you would be entered. The second contest was to write an essay, and we had a question, and we got a bunch of entries, and the winner, we selected it by grading it, because what do? We grade things, so we did that. And the third one is just to go to a website and fill out a very short questionnaire. Drobo, or Data Robotics, is really interested in whether a podcast audience is a good place for them to be. So they'd like to know what the listeners are like. So there are about 20 questions. Multiple choice, really easy, no essays, no fill-ins. It's really easy to do.
You will not be graded on your answers.
Absolutely, no grading. You can go to bit.ly/TWiVDrobo, or you can find the link in our show notes. And in about 2 weeks, Data Robotics, Mark, will randomly select a winner. So you should just go check that out. We have some good responses so far, but make Mark happy. Go and check it out, and then you'll be automatically entered. And Kirsten, you can enter too. You can just go over there and enter the contest. But Drobos are great. There are a whole bunch of them. I'd suggest starting out with a 4 or 5 drive model. Rich and Alan, you guys should get one at some point.
Yeah, it's on the list.
You could fill out the questionnaire. You're not a family member, so you can enter the contest as well.
Oh, so TWiV hosts can enter this.
You can enter it. Yeah, sure. Go ahead.
Oh, I'm all over it. I assumed I was disqualified.
Nothing in the rules.
Yeah, I had been operating on that assumption for all these contests.
No, the first two. Yeah, the first one or the second one when you had to answer a question and get graded, that's probably a conflict. But the others, do this one and see if you win. And of course, as always, we thank Data Robotics for their support of teaching people about viruses, and that's what TWiV is all about. We have a story that just came out this past week. It was sent by Srinivas. It's called Smallpox Vaccine May Protect Against HIV. And Srinivas wrote, "Can you please comment how much of this is true?" Which I thought was an interesting comment. It's an article in, what is the journal? Biomed Central. Immunology, right. So it's a science article. So I think it's funny that he says how much of this is true. But it's an interesting problem, and this is right up your alley, Rich.
So what Srinivas actually wrote in about was this Washington Post story.
Right.
It's about a 4-paragraph deal, datelined May 19th. The headline is, "Study suggests smallpox vaccine may protect against HIV." And there's a subtitle that says, "End of smallpox may have led to HIV rise." And the science in it it's only a 4-paragraph story. The second paragraph is a single sentence that contains the science that says, "Laboratory tests suggested that immunity to smallpox triggered by the smallpox vaccine can inhibit the replication of the AIDS virus," researchers said in the journal BMC Immunology. And beyond that, there's that story, which we can discuss, and about 2 or 3 other parallel stories. And I'm not sure they collide, but they're certainly parallel. One that you found, Vincent, which was the original PNAS story that is titled "Evaluating Plague and Smallpox as Historical Selective Pressures for the CCR5 Delta 32 HIV Resistance Allele." So that PNAS story was asking the question, why is there in the European population a relatively larger percentage of people that are defective in this CCR5 receptor? And the common theme in this whole thing is the CCR5 receptor. So this is a protein that's actually a chemokine receptor. Chemokines are signaling molecules in immunology. So this is a receptor that sits on the surface of cells and senses the presence of chemokines and sends off other signals in response that influence the immune response. And that molecule is used as a co-receptor for HIV for entry into mostly macrophages, if I understand it correctly. HIV uses CD4 and CCR5. And importantly, there's a couple of different kinds of HIV. One that uses the CCR5 receptor, and that's the one that is usually responsible for the initial infection in a person. Okay, so there's a bottleneck in the HIV transmission from person to person, in that usually the first HIV is what we would call CCR5 tropic. That is a virus that uses that co-receptor. And as the virus, we've said, it's very variable. As it evolves in an individual, it throws off viruses with other tropisms, in particular CCR4, which is a different co-receptor. But in the transmission process, you seem to select out the CCR5 tropic viruses again. So there's a population in Europe, we've discussed this before, that are defective in this CCR5 receptor, and since that's important in the transmission of HIV, those individuals are resistant to HIV. And so the question becomes, where did they come from? Why is there such a large proportion of them in Europe? And this PNAS paper that you found suggests that just using mathematical models and population genetics, suggests that somehow smallpox may have selected for those people, as if survivors of smallpox, you had a greater chance of surviving smallpox if you were one of these individuals. So that's where that puts out there the idea that there may be some relationship between smallpox and HIV. And that's where the paper behind this Washington Post story starts. And what they describe in their introduction is they say, HIV, according to the models, entered the human population decades before it actually burst out in the open and really bloomed. Okay, so it was probably simmering somewhere at a low level, and then all of a sudden it really blossomed. And why was that? And the postulate is that maybe it's because of an interaction with some other pathogen, and maybe that was smallpox. So smallpox was declared eradicated in 1978, depending on 1978 is good enough. And at the same time that it was declared eradicated, the World Health Organization recommended against vaccinating any further against smallpox. So up until that time, you have a population that is immune to orthopox viruses, either because they had smallpox or because they'd been vaccinated. And now all of a sudden, the population is no longer exposed to smallpox or the vaccine. And coincidentally, there's this bloom of HIV. And so the question is there a relationship? What these people did in this paper is simple. It's just a couple of experiments. They took two groups of people, vaccinated some of them with the smallpox vaccine. Some were not vaccinated. And then they isolated from them what are called peripheral blood mononuclear cells, which are basically macrophages. And they tried to replicate on those cells HIV that was either Ccr5 tropic or Ccr4 tropic. And what they report is that the replication of the Ccr tropic virus is reduced in people who are vaccinated relative to people who weren't, whereas replication of the Ccr4 tropic virus is the same in both populations. And so here we come back again to this Ccr5 thing, and maybe the Ccr5 is involved. And so what they're using this as support for the notion that maybe smallpox vaccination or smallpox infection itself somehow conferred some resistance to HIV that was specific to Ccr5.
So you have smallpox out, HIV in.
Right. So, as I said, there's a lot of parallel stories here. You know, the selection for the Ccr5 negative individuals doesn't totally jibe with this in my mind. Right. I can't see the connection. And I also, the other way that Ccr5 shows up is that there was another paper, the senior author is Grant McFadden from 1999, a science paper showing that Ccr5 and other chemokine receptors assist in poxvirus infection. So that idea is not new either. They originally thought it might be a co-receptor. Now it turns out it's because of the virus tweaking that receptor, and it modifies the intracellular environment so that it replicates better. So there's a lot of parallel stories here involving HIV and Ccr5, and frankly, I have a tough time putting them together, and I don't know quite what to make of the magnitude of the response that they see here. And I noticed, Vincent, that you had some interaction with Scott Hammer about this.
Yeah, so the data in this paper are basically, they're virus infections, as you said, blood cells from these two sets of people. They infect with HIV, and then they measure virus that are released from the cells. And the way they do that is to measure an enzyme in the virus particles. So they're not actually measuring infectious virus, but rather an enzyme that is in the virus particle. And there's really a 5-fold difference between people who were or were not immunized with the smallpox vaccine. And to me, I mean, that could be, 5 times as much virus, or it could be 2 times as much virus. There's no way of knowing. So I asked Scott Hammer, who is a HIV guru here. He said, I'm not really sure what to make of this report. A 5-fold lower reverse transcriptase level, that's the enzyme they're measuring, seems modest to me. In HIV-infected people, a 5-fold lowering of plasma viral RNA is clinically meaningful with respect to delaying the time to clinical progression, that's AIDS or death, but it's still modest. And he says we like to look at interventions like drugs or vaccines that lead to a 10-fold reduction or more. So it's not a lot, but it's real, right? And
of course, a very, very modest effect on a big population over a long period of time it gets amplified, all right?
Yeah.
So, I kind of agree. There's a mystery here to me. First of all, okay, so the geek in me says, first of all, they're reporting an effect, okay, and you can't deny that. They see a 3-fold difference. They've done all the 3 or 5-fold difference. They've done all the statistics, everything else. They got nice controls with the Ccr5 virus and all that kind of stuff. So there seems to be an effect. I would like to see that reproduced, okay, by somebody else, somewhere else. There's aside from all of these hints that CCR5 keeps popping up in poxvirus and HIV infections, and there's some commonality there. Mechanistically, I don't understand. It doesn't compute that a smallpox vaccination would throw off macrophages that are somehow, I suppose you can say that it somehow down-regulates CCR5 or something like that. Mechanistically, I can't make any sense of it.
Well, you could be surprised, Rich.
Yeah, I could be surprised. And I must say that it is a huge leap to go from those cell culture studies to smallpox vaccine may protect against HIV, or the end of smallpox may have led to HIV rise.
Yeah, it's hard to find the weak link in this study because there are so many to choose from. But as I looked at it, a whole bunch of things jumped out at me right off the top. It's an interesting hypothesis, and this is certainly an interesting experiment and worth doing because it's so straightforward. It really doesn't put anybody out to do this. But what do we really know after this? And there's a big problem going from the data to the hypothesis just in terms of dates, because smallpox vaccination ended worldwide around 1978. By 10 years after that, we had a full-blown outbreak of HIV in multiple countries around the world among adults.
Yep. Right.
Now, how do you explain that if this is due to these people? The only people who were not vaccinated against smallpox in the 1980s were people who were too young to have received the vaccine.
Good point. Very good point.
So, and they were not the ones showing up as patients. You were getting people who were middle-aged, who were adults. And this is not, there's no dynamic that I've seen in any of the data from the epidemic that would show that this skews toward people who are, below the cutoff for smallpox vaccination. So it doesn't line up with the epidemiological data. And then you've got all this question about, I didn't know the full CCR5 story. It seemed kind of confused to me, but now I see that was not an illusion.
Like I said, there's about 3 parallel stories that they don't intersect.
Right. In my case, I always, when I see something and it confuses me, I say, well, I'm not that bright and I'm not up on this field. So that's a possibility. But then the other possibility is that maybe it's just really unsettled. So we have this open question around whether CCR5 is what it's doing in this case, if anything. And then the effect that they saw, I mean, a 5-fold reduction. I don't know. I like to see things happen in logs in
yes,
in cell culture. When you're doing a cell culture experiment, you can get massive results, and it seems like you're really kind of, in this case, almost trying to read the data in terms of the hypothesis rather than in on its own terms.
So, Alan, where does the Washington Post get? How does this story wind up in the Washington Post?
2 words: press release.
Yes.
Right.
And in fact, what you're looking at on the Washington Post site is this is kind of tertiary news. So they cite the McClatchy Tribune as the source, and what that means is this is a wire story. The Post is picking up articles that have come out from all over the place and republishing those as content, so visitors to the WashingtonPost.com or people who still get the Dead Tree edition, that dwindling population, can pick this up and get news from around the country without WAPO having to have their reporters all over the place. So that produces these little stringers, these short little blurbs. This story apparently appeared in the McClatchy Tribune, was then picked up by the Washington Post, was probably picked up by other places. And it's just tantalizing enough that a news editor who's on a daily deadline would look at it and say, sure, throw that in with the health stringers.
Okay.
How it gets in the McClatchy Tribune, somebody is sitting there looking at what I look at every day, which is hundreds upon hundreds of press releases generated by all sorts of research organizations and journals. And anybody who's publishing a paper these days seems to publish a press release saying, here's why it's newsworthy, whether it is or not. And depending on how well those are written, they can sound very persuasive.
So this originates, the ultimate source is the scientists themselves.
It's the press office for the organization, the institution.
Okay.
Yes. So your institution, whether you know it or not, Rich, has a press office.
Oh. Are they watching me?
They may be aware of TWiV, or they may not. But if you're doing a research that you mentioned to your school is interesting, they may send somebody from the press office to talk to you about it, and they might write up a press release. Or you may send a paper to a journal, and the journal will write up a press release to go out with it. In fact, my very first journalism work as an intern in the Nature offices in New York was writing their press releases. So all of this is put on the hook as bait to try to get coverage of the journals, to try to get coverage of the researchers, and no objection to that. I mean, it's a major source of information. It's very useful, even if there's an awful lot of it. But the trick is to separate the signal from the noise, and in this case, that process got a little derailed.
So, but it is possible, if not common, for the person who actually did the work, right, to bring this attention to the attention of whatever.
Yes, absolutely.
Yes, absolutely. I mean, if you look at something like the publication we just saw the other day from Craig Venter's Institute, Venter, I'm absolutely certain, and he's on the video. He's front and center promoting his work.
Right.
So a lot of researchers are very promotional of their work, and again, no objection to that. That's something that I think probably more scientists should be a little aware of. But the issue is that when they're promoting it to an audience of science journalists, many of whom don't have a very strong background in science, they just ended up in this field because their job evolved, they can sometimes be misled. So the people crafting the press releases have to be kind of careful about how they do it and balance that with their desire to promote interesting work.
One of the reasons that Kirsten's job is so important, and our job as well, and that is to make sure that as many people understand the science as possible, so that they can pick the wheat from the chaff.
Mm-hmm. Yeah, there's a certain point though where sometimes the sensational headlines, I feel like there is a certain amount of, sensational headline will grab attention, but then if you can actually write the article so that the information is in there, in a way that does justice to the science, then you're kind of balancing it out. But there's a certain point where, what is the job of the news and the media, and what are you actually putting out there? This story in itself, I don't think it's going to, change the way that people think about anything.
I'm worried a bit because, people are very sensitive to vaccination. And here the article is suggesting, and the article itself it doesn't give you any detail. So
yeah, true.
People really can't make a decision based on it, but they'll say, eradicating a disease is bad or good? I don't understand, right?
Right. Well, we can now, I think, do the study now that smallpox is there's a resurgence of it in certain areas of the world. Maybe we can look and see what happens to HIV levels in those areas.
Actually, there's not a resurgence of smallpox. There's some other pox viruses crop up here and there. Monkeypox.
That's an interesting question. Maybe some of the animal pox viruses would do a similar thing in an animal.
Right.
You could look at that. But I think that this will stimulate some more work, which would probably not be a bad idea because it's an interesting finding. Yeah. But the implications for HIV are very vague, and I think, Alan, the timing just doesn't make sense.
No.
Yeah, that's a good point.
The dates don't line up.
In fact, if they want to...
Yeah. Go ahead.
If they had just said, this is the observation, for the virology community would have had as much of an impact. But then to go and start the paper by saying maybe this led to HIV emergence, I think is pushing it a bit. But
yes.
Rich,
so I just wanted to tag onto this a bit that we missed last week. We record these on Fridays, even though the rest of the folks don't get them till later. But last Friday when we were recording, May 14th was Smallpox Vaccination Day, the anniversary of the first smallpox vaccination in 1796. So I've got this on my iPhone now, so I'll never forget it again. May 14th.
You got a 4-day notice, a beep ahead of time, right? Well, that's it. Well, 1796, that's when Jenner first did it, right?
Right.
But I think
that's when he did his first vaccination.
Yeah, I understand the Chinese were doing it for thousands of years before that, though.
Well, this is the first practice of what we would call Jennerian vaccination, where you actually take cowpox virus and administer it by scarification. There were lots of other immunization practices leading up to that.
Yeah, I think the Chinese used actual virulent smallpox virus, and
that's correct.
And some people died, but the ones who didn't were immune.
What doesn't kill you makes you stronger.
That's right.
Exactly. And that was followed up by the Chinese did it by crushing up crusts from smallpox victims and blowing it through a silver tube into the recipient's nose. Okay, I think that was a pretty dangerous technique. And that was followed up later in the Middle East by a technique called variolation, where people took stuff from active smallpox cases, a little bit of the pus from one of the pustules, and scratched it into your skin. And you got a really pretty nasty reaction to that. You were contagious for smallpox, but the case fatality rate from that was 1% or less, whereas the case fatality rate from a natural smallpox infection was 30%. So you were increasing your chances of survival by 30-fold, and since you're pretty likely to get the disease, that was worthwhile, and that was practiced all over Europe and in the Western world, in the North America, for a long time before Jenner came up with basically a safer method using a related virus, cowpox, instead of the actual smallpox.
All right, well, happy smallpox anniversary.
Thank you so much.
Because you work on it, so
I'm just really happy about this.
All right.
I had something. I know this is not on the rundown for today, but did you guys see the Ebola vaccine news that came out of the NIH?
I did not.
I did, yeah. You know why I saw it? Because you just tweeted it. I hadn't yet seen it, but yeah, I went and looked at it, and it's an experimental vaccine, and it protected monkeys, I think, right?
Yeah, just monkeys. And it protected, it was a vaccine that was developed to protect against some of the older or the original forms of the Ebola virus. And they tested it recently against also a newer form of the virus, and it gave protection against that as well. So what they're hoping to be able to get out of it is a universal Ebola vaccine.
Yeah, this is actually an interesting strategy. It mimics a lot of the vaccination trials for HIV, and that's a 2-component immunization where the first one is a DNA vaccine, which encodes one of the viral proteins, and then you have a boost with an adenovirus that carries a gene for one of the viral proteins as well. It's called prime boost, DNA prime and adenovirus boost. And that seems to have some promise for HIV and now for Ebola.
And maybe for smallpox. No, sorry.
Well, we have a pretty good vaccine for smallpox, although it probably could be improved. We've talked about that quite a bit on here. And
there are a couple of other newer versions, recombinant versions that have been worked on, and that look quite good.
Ebola is particularly tough because it's a dangerous virus to work with.
Right.
And I'll bet in that study they probably used the Reston strain, which doesn't kill people. That would be my guess in the monkey challenge experiments. Otherwise, you have to do it under incredible containment, and it makes it very difficult to do that. But that's very cool, yeah. We have one more story about an RNA virus that I thought is very cool. It's a bit geeky, so bear with us. As everyone knows who listens to TWiV, viruses can have either DNA or RNA as their genetic material. And the puzzle is the following. DNA viruses can have huge genomes. Over 1 million bases is the biggest known viral genome, 1.2 million base pairs. And then the viruses that have RNA genomes are pretty small. The biggest ones are coronaviruses like the SARS virus. They have 26, 30 kilobase genomes. They don't get any bigger than that. And the genomes of most RNA viruses are small. And we think one of the things that limits that is because RNA viruses make more mistakes when they duplicate. So all the enzymes that copy nucleic acids make mistakes, but those that copy DNA can correct their mistakes. They have what we call proofreading. And RNA viruses don't have this in general. But people have always wondered about these coronaviruses, these virus family that includes the SARS virus, because they're so big, 26, 30 kilobase genomes. And these viruses encode for a lot of proteins, and among them are proteins that look like they could correct errors, or in other words, be involved in proofreading. And this paper, which came out of North Carolina and Nashville, basically looks at one of these viral proteins in the SARS coronavirus. It's called NSP14, so a name that means nothing to most people, but to us it means non-structural protein number 14. And it had all the signatures. If you looked at the amino acid sequence, it looks like an enzyme that can cut RNA from the ends. So that might be something involved in proofreading. So what they did in this paper is to just remove this gene from the virus. And they found that the virus is actually growing reasonably well without it. It doesn't grow as well as viruses that have the gene. But the virus makes a lot more mistakes than when it has this protein made. Okay, so they do massive sequencing. They grow the viruses and pass them in cells and do a variety of different selections. And they basically show that if you take away this NSP14, the virus makes at least 10 times more errors than it normally would. So this means that this protein is involved in error correction. It's the first time that we have such a protein in an RNA virus. And it's probably why these coronaviruses have such big genomes. So I think this is really cool, and for mostly virologists will appreciate this, but for all of you who are not, this is a really neat thing because we never knew that RNA viruses were able to have erasers on their pencils.
Yeah, it's interesting. It's a general property of RNA polymerases, anything that makes RNA. That's including the virus replicases and the normal RNA polymerases that make messenger RNA in cells that they can't proofread. And it's almost like evolutionarily. You got to a point where you're making bigger and bigger genomes, and you get to a barrier without that proofreading, and without adding this as basically an accessory function to clean up the mess, you can't get any bigger. And once you do that, you break through that barrier.
Yeah.
Very interesting.
That seems to be the limit, about 30 kb. Beyond that, you make too many mistakes, and it's not compatible with making a virus. But the fact that these viruses live without this protein, or I shouldn't say live, they can replicate without this protein, means there must be some other mechanism, right, that they haven't found yet, because it's obviously not making so many mutations that the virus can't grow.
Well, I don't know if they did there must be enough data here to do the statistics to figure out whether the mutation rate that they're seeing is consistent with not having any proofreading, because there are a fair number of experiments on error rates in RNA viruses. I didn't see whether this thing was down around, the normal error rate for just an RNA polymerase.
I think it's within the range, but they do say that they can't conclude that this is the only correcting protein based on
there's a lot of other
so
NSPs in this virus that they don't really know what they do.
There are a lot of other proteins. So there's plenty of work to do here, but as a guy who's worked on RNA viruses the whole career, I think this is very neat to really understand how these genomes can get so big. They have to fix their errors. So
one of the things I like about this paper is it's got everything in it. It's got plaque assays, it's got one-step growth experiments, it's got mutation rates and all of that kind of stuff. When I teach I teach the introductory lectures in our advanced virology class, and I try and teach all of those basic virus-specific techniques, and in the context of I try and teach them genetics at the same time. So last night, I put this in my folder for a teaching paper for the coming years, because I think this is just really great. It's a really nice piece of work.
It also has deep sequencing.
Yep.
Everything.
It's a wonderful meld of all the classic techniques like plaque assays and one-step growth experiments, and the newer stuff like reverse genetics, deep sequencing, all the rest of it, to illuminate a new principle or a new twist on an old principle of how you deal with mutation rates.
Right.
It's great.
Great. Now, Kirsten, you have to get out of here soon, right?
I do. I am going to have to run. At noon, I have my Justin TV program.
Can you listen to one email?
Absolutely. I've got about a little less than 20 minutes still, so I'm good.
Because this first one, I thought you would have a particular opinion on. I actually saved it from last week so you could listen to it. This is from PJ, and the email was entitled "TWiV Gets Weird." And this was because we had an episode called Darwin Gets Weird. And he says, well, not really weird, but allow me some criticism. I just listened to the Darwin Gets Weird episode, which was, again, a real treat. Thinking about prions as non-Mendelian inheritance factors seems logical, albeit the question is, how much information is carried by them? Do they belong to the general assortment of stuff in the zygote, which, with its chemical gradients, also carries some information like orientation, or should they be considered a third factor? So I don't know if you know about this story, Kirsten, but prions have been shown to mutate. And, these are just proteins, but you can select them with drugs. So
yes. And when they mutate in a bad direction, you get mad cow disease and Creutzfeldt-Jacobs and those sorts.
So I don't know. What do we think? I mean, he's talking about epigenetic inheritance here. How would you classify a prion? I don't know.
Well, as far as I know, prions they're all across the animal kingdom in cell membranes, from yeast to humans in all sorts of places, and having, the function really has not yet been clearly elucidated, has it? I mean, I really don't think so.
Well, the one in people hasn't been. We don't really know what that does normally. In yeast, they have some functions in their prion state associated with them. So,
right, and they're usually, and they seem to, as far as I recall, they're related to like copper metabolism, or there's something to do with the metal metabolism.
Yeah, so they let yeast respond to stressful environments.
Yeah.
So if you put heavy metals in or remove a nutrient, the prions can change really quickly, faster than you could make, say, a new gene product, and they allow.
Which in the case of yeast, if you want some kind of rapid adaptation, or even with yeast where they have a rapid life cycle, and they would be dividing very quickly, then you'd have an evolutionary mechanism. I wonder how that would hold over into into the mammalian sector, or longer lived organisms. Would that still possibly have the same evolutionary effect? Yeah, I think that's a really great question, and I hadn't heard it before, but that's really interesting.
I don't know. I would probably consider them as epigenetic, but that's only because we define genetic as nucleic acid. So
right,
by definition,
it's a little tautological.
Yes, exactly. But no, as we learn these new ways of having inheritable changes, you have to expand your definitions clearly.
If it's not actually, a lot of epigenetics also is the way that various factors affect the genome, and I don't know that necessarily these prions would be affecting the genome.
No.
So in that respect, they're not necessarily epigenetic, but they would be a third factor.
You're right.
So we can argue it.
They are not affecting the genome. You're absolutely right. Like methylation of histones would affect the genome expression.
Right.
Yeah.
Right.
Maybe it is a third factor. Well, PJ continues, however, and this is why I use the TWiV Gets Weird headline. You've ventured into an area that I'm rather passionate about when you mentioned a possible TWiV iPhone app. This may sound a little strange, but the whole Apple I ecosystem is built in such a way that it keeps stuff in. That is, it's a closed system. A native iPhone app will only run on Apple's iPhone, iPad, and iPod, so everybody wanting to use it would have to buy one of those products. That's quite the opposite of what I call open access. Luckily, there's an alternative that has been invented by Tim Berners-Lee a while ago called the web. Data available on the web is usually open access by definition, unless the information owner wants to monetize or protect it. Now, a native application has the advantage that it can store data locally on the device, and here's the good news. HTML5 on most browsers, WebKit as used on the iPhone and Safari, but also as part of Google's Android and Palm's WebOS, and Google Chrome. Firefox should not be far behind. Have everything you need to write applications using JavaScript and persistent storage methods using a local database. Those applications are basically websites on steroids that can work offline. They are by their very nature cross-platform. You can distribute them without having to go through iTunes or Google Android Store. Use them both on mobile devices and on the desktop. I've been an open source advocate for many years now, and the trend towards closed platforms away from the web, or euphemistically walled gardens, seems worrying to me. Many people seem to be willing to trade freedom of access for ease of use, even though this trade-off is not really one we need to make. So if you do make such an application, please don't feed it to a closed platform. It would be a pity to have the data just there. Thanks for the show, PJ. So you understand this, Kirsten?
Yes, I do. I'm an Android user myself. I like the open platform, that effect. And going Android, it helps you go viral that much more easily.
What would be better for TWiV, right?
Exactly.
Well, I had thought of making an iPhone app about viruses because I think it would be great for people to carry around in their pocket a database. If they hear about a virus, they could just tap on a word and go learn all about it. And I think this is fine to keep it open sourced and outside.
Absolutely, it spreads it around more.
And
more
of course, if you write
write
a web app, it can be used on an iPhone as well, right?
If you write a web app, it is used and useful on an iPhone. The Android platform, though, is interesting in that if you program something for the Android platform, it's only a couple of short, simple changes in coding, I guess, to adapt it for the iPhone. So it's also something that if you go open Android first, and then you can go to the iPhone.
Right.
And then if they let you in.
Yes, right. They have to let you in.
Yeah, you have to stimulate their receptors appropriately.
And not stimulate the wrong ones.
Right.
Well, we'll do that, PJ. I appreciate the advice. We will try and keep it as open as possible because it has to go viral. You're absolutely right. And Howie writes, awesome podcast. This is science education at its very best. I'm a mathematician who has started to work on population modeling. I listen to both podcasts regularly and have watched about half of your course lectures. 2 comments. 1, for bacteria transferred from one medium to another, there is an initial lag phase where the individual bacteria are synthesizing RNA, enzymes, and other molecules they need for growth prior to their resumption of division. Then comes the exponential growth phase, which is eventually limited by the exhaustion of available nutrients and accumulation of inhibitory metabolites or end products. Thus, at first, the bacterial population is not growing. So I think that's in response to a show where we're talking about the difference when you put a bacteria in medium versus put a virus on cells. The virus doesn't have a... It can't start reproducing immediately because it has to make the parts. Whereas a bacteria, after a little bit of time, it could just divide and make more bacteria. So that's called the lag phase. Right. And then number 2, the mathematical epidemiology world seems to believe that pathogens evolved to increase their fitness, measured by their RO, basic reproductive rate, thus to become more easily transmitted, make individuals sick longer, and to decrease virulence, host mortality. I sometimes get a little confused by your discussion of this. Oh dear, we don't want to do that now.
No.
Well, it is confusing.
Well, it's not as
because we'll say sometimes generally viruses evolve to be less virulent and be and in equilibrium with their hosts, except that the motivation really is transmission, and sometimes transmission involves a pathology that we experience as virulence, something nasty, and so the virus may want to do something nasty to us, like create lesions all over your skin or something like that, or give you raging diarrhea in order to spread itself. Okay, so transmission, I think, is the key.
I agree with you. The problem here, and I think the key is, he says the mathematical epidemiology world, and that just isn't the real virus world, right? Mm. That those are theoretical models that simply don't always aren't always right, because viruses, you're right, we think they evolve to be transmitted most efficiently. In some cases that means more virulence, in some cases it doesn't. There are plenty of examples of infections where we don't get sick, right, and they're very successful viruses. So that's why we may be confusing him, because there are differences, but that's the way it is out there.
Right. The issue is that you can't have one mathematical model that will explain the evolution and virulence of all viruses, because they choose different strategies.
Right.
They can choose a strategy that leads toward decreased virulence and chronic infection, and that is one way for a virus to survive. Or they can evolve, I shouldn't say choose, or they can evolve in a completely different direction depending on their transmission route and increase virulence so that they rapidly spread from host to host.
Right. Okay.
This is actually one of the things that I emphasize in teaching virology all the time, is that one of the really fascinating things is how much variation there is. There are very few real solid principles that apply to every virus. Every virus has to have a messenger RNA somewhere in its life cycle, and beyond that, it's all over the map.
Yep.
Right.
Okay, Howie, thanks for writing. Howie's a professor of mathematics. It's great that you're listening to TWiV. Very good. Yeah. And we have Kara writing, as I was listening to the episode of Futures in Biotech that Vincent was on, I had a question. There was a philosophy towards antivirals that you discussed on the show. Generally, antivirals should be used very cautiously, avoiding monotherapy. I completely agree with you in the case of HIV and HCV and viruses that cause infections like those. That is the only way to go in that case, but I don't know that it is as practical current drug development market for other infections. My question is about Tamiflu and the other influenza drugs with similar targets. Isn't the fact that influenza is a self-limiting illness the reason we do not attack it with multiple drugs like we do with HIV? In an influenza infection, our bodies can often clear the flu without drugs, and we come out without any lasting ill effects. HIV and HCV cause chronic diseases that ultimately kill their hosts. Thank you for doing this. Okay. So what do we think, guys? My view is that we would love to have multiple drugs for flu so we don't get resistance. Right. And that is why we don't have them. But, you know, in the past year, people were talking constantly about our anti-flu arsenal is too small, and that's why we get resistance immediately to Tamiflu.
Yeah, keep in mind that flu kills, in the United States alone, 30,000 people a year. So it is not an innocuous disease.
Yeah.
And the more tools we have against it, the better off we are.
And an issue with the flu drugs is they're relatively new, and Tamiflu and Relenza, for example, come from different companies. So there's not a huge incentive to have combination therapy from that perspective.
Mm.
Even though it may make a lot of biological sense, what you see is marketing of separate drugs.
Yeah, and they have to also be tested together in combination to make sure there aren't any.
Yes, you'd have to make sure that they're not going to. I mean, it's unlikely since they're working by similar mechanisms, but you'd want to make sure that they're safe together.
But there are multiple new flu drugs in the pipeline, so I think we will see combination therapy in the future. But remember that HIV and HCV go on for years. You have plenty of time to diagnose and then treat the infection. With flu, you have a few days, and if you don't catch it, then it's too late to treat. So that really limits your options there.
Actually, I just learned the other day, too, that clinically, the way they manage HIV now is that not necessarily to jump on it right away because the drugs are so toxic. They wait. If you're diagnosed with HIV, they wait until until there are issues, and then they start to jump on it with drugs.
Yep,
it's very interesting.
That's right. Hey, Kirsten, you want to leave now? Or did
I should probably head out very soon.
I don't want to kick you out.
Email?
I just one more. Sure.
It's getting to the end.
We got just one more. Yeah. Okay, this is from Levi, who probably Rich knows.
I do.
I'm a grad student at the University of Florida studying HSV-1 latency. Advisor is Dr. David Bloom, and I'm lucky enough to work next door to Rich Condit. He recommended TWiV during our spring virology journal club, and it has often been a source of material for our discussions. I wanted to give a little something back by suggesting you have a look at a film preview for a documentary about HIV/AIDS. So as an avid moviegoer, I frequently go to Apple Trailers to watch movie previews. While viruses in films are often subject to extreme artistic license, I recently saw a trailer for a film called House of Numbers that may make a more serious error. It's a documentary about HIV and AIDS, and it supposedly includes interviews with many of the heavy hitters in the field. Unfortunately, the preview for the film had a heavy overdose of AIDS denialism of the kind promoted by Peter Duesberg and others. I have not seen the entire film, so I don't know if it really pushes this agenda, but I hope not. The attitude of AIDS denialism can only hinder our efforts to counter the AIDS pandemic. As you have mentioned in previous episodes, there are controversies and conspiracy theories surrounding many diseases, viruses, and vaccines, perhaps none more than HIV and AIDS. Hopefully this film takes an honest, objective look at HIV research and is not just a piece of sensationalism intended to stir up controversy or enhance a filmmaker's career. I will try to get my hands on a copy of the film so I can judge for myself. Perhaps the TWiV team can do the same, and he provides some links which we'll post. According to the Wikipedia article, it appears that my fears may be well-grounded. You can judge for yourself. These controversies and conspiracy theories can only be fought with quality research and education of the public at large. Hopefully, virologists like myself and those of the TWiV team can be a part of the process of separating fact from fiction. Look forward to meeting you during your visit to UF this summer. Thanks for the podcast. Well, I looked at these, and it seems to me that they are, the Peter Duesberg school of AIDS denialism.
That's too bad.
What was his phrase here? I don't know. Look, it looks pretty nasty to me.
Yeah, from what I saw, they're not really balancing it. It's mostly the denialists talking, and I don't think it's useful to give them a lot of time because we know.
Well, this is the downside of everybody having access to the tools of media making and distribution.
Yeah.
Yeah, but the fact that there are so many routes of access, there are many ways to fight back. So
yes.
I know with the what was the film, the creationism film that attacked people. I don't remember which one it was. A couple of years ago that came out. Anyway, through new media, people struck back, and I don't know that the movie actually made much of an impact at all.
Well, maybe we can. Go ahead, Rich.
Levi's quote here is. He's hoping it's not just a piece of sensationalism intended to stir up controversy or enhance the filmmaker's career. From what I can see from the links he gave, it looks like just that.
Yeah.
Sensationalism. And it looks like they've taken a lot of comments out of. I mean, you can a legitimate scientist, if you get him into a room and talk to him for long enough, you can get him to say, "Well, I'm really not sure."
Yeah.
Okay.
That's right.
And if you just cut that out and stick it on the front page, you can make a story out of it. But that's not real science or journal.
Right. Or you can set traps like, isn't it possible that?
Yes. Right.
Yes. Well, of course it is.
Mm. Well, thanks for sending it, Levi. We'll post the links, and if anybody has questions, just send them in. Now, usually we do some picks. Kirsten, do you want to stick around or do you want to cut out?
At this point, I should probably run. It is now noon, so I got to jump over.
I want to thank you for joining us today. It was great.
Thank you so much. I've really enjoyed it. This is, your program is fantastic. I'm a big fan myself, so it's been wonderful to actually get to sit here and talk with you guys.
Come back again.
Thank you. I'd love to.
Thanks, Kirsten. It was great.
Thank you.
Where would you like people to find you?
You can find me if you Google Dr. Kiki. Use Google, Dr. Kiki. I'm all over the place. Or you can go straight to my website, which is kirstensanford.com.
Thank you. Take care. Have a great Q&A.
Great. Thank you very much.
All right. Bye-bye.
Bye.
Bye.
Okay, guys, let's do some picks of the week. Alan, what have you got?
I'm sorry. I was concerned that when she left, the whole call was going to end.
She's still there.
I'm still here. I closed Skype, but the call didn't go away.
Sorry.
Oh, okay.
Oh, that's interesting.
Yeah, that's a good question. I am in charge, am I not?
Oh dear, you are in charge. Okay,
you're the queen of the call.
You can close it now.
I will not leave.
It'll just take us a few minutes. We're doing some picks.
Yeah,
I'm sorry. Alan, what have you got for a pick of the week?
My pick of the week is just a handy little tool that I found a couple of months ago called Evernote. And it's a website that functions as a notepad. So if you're away from your computer, but you have access to a web terminal and you suddenly need to jot something down you'd really like to refer to later, you can just log into this site. You set up a free account. And when you sign in, you can take notes. You can jot down whatever you need. It's just a little minimally functional word processor type window. And then you save and exit. It's a little like Google Docs, but it's just very simple, very straightforward note-taking. And maybe not for everybody, but I've found it handy.
It's a great app.
I might just try that
Great
out.
app. And it's also available for iPad or iPhone, I believe.
And since it's on the web, you can access it through any of those devices.
That's right. It's not a walled garden. Exactly.
That's right.
Okay, Rich.
So last week I chose a book that was given to me by my daughter and son-in-law because I'm a science geek. Actually, there were two books, and this is the second one. It's called The Knife Man, Blood, Body Snatching, and the Birth of Modern Surgery by Wendy Moore. And this is a biography of a famous surgeon named John Hunter, who was Scottish, lived between 1728 and 1793, and he's credited by many as being sort of the father of modern surgery. He was an anatomist and created a lot of surgical procedures, and he was a very interesting dude as well. I mean, he had a house with something like a fort that was basically a museum with 14,000 display pieces in it or something like that. So this is a wonderfully written and very interesting story about the history of surgery, basically.
Nice.
And an interesting character.
Something I don't know much about.
Oh, I recommend it.
Very good. Well, my pick is a blog. Now, back on TWiV 42, we had picked Effect Measure, another blog, which is a forum for public health research. And that is a great blog. It closed down this week. But the writer has promised to post over at The Pump Handle, which is another blog over at scienceblogs.com. It's a water cooler for the public health crowd, and really a great blog for anyone interested in public health and the environment. And if you're interested in that, you'll know where the name The Pump Handle comes from, of course. You got it there, Rich?
It. Yes.
Yeah, I got it.
All right.
Took me a...
And that'll do it for another TWiV. You can find us on iTunes, at the Zune Marketplace, even through Stitcher, the app for your mobile phone. And, of course, you can always go to twiv.tv to listen and see all of the show notes. And as usual, please send us your questions and comments to twiv@twiv.tv. Alan is at dovedocs.com. Great to have you back again.
Great to be back.
Rich Condit down at the University of Florida. Professor Rich, I'll see you in a couple of days.
Looking forward to it.
All right. And Dr. Kiki, great to have you.
It's been a lot of fun. Thank you.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.